Next, we're going to cover the culture media that we use to grow mycobacteria. Mycobacteria are strictly aerobic. They grow well at high humidity. So here in the Cleveland Clinic, we use 50 to 70% humidity in our incubators. We also use about 8% CO2, and we use body temperature at 37 degrees Celsius for growth. There's 3 types of media that we use: an egg-based media, which is Lowenstein Jensen, an agar-based media, which is Middleburg 7H11, and a liquid media in our midget tubes, which stands for Mycobacteria Growth Enhancing Tube. This media is Middleburg 7H9. It's important to use multiple different kinds of culture media so that you can grow all different kinds of mycobacteria. If one is suspecting Mycobacterium pomaphilum, it's also important to add a hemin disc to your midget tube so that it can get the appropriate heme cofactors for growth. Once we have found positive growth in the midget tube will indicate that to us by fluorescing, which occurs once all of the oxygen has been consumed within the tube. So in order for that to happen, we make sure that all of the tubes are tight so that no extra environmental oxygen can be introduced. All other culture media, we make sure to loosen the cap slightly so that aerobic oxygen can come into the culture and allow for growth. So once our midget tube has been flagged as positive and we stain it with the Ziehl-Neelsen stain, confirming the presence of acid-fast bacilli, we will take a small sample and plate it onto a blood agar plate, which is what is used commonly in the bacteriology lab. This can check for contamination and is also good growth for rapidly growing mycobacteria. 7H11 is our agar of choice, which is what we use mostly. This is just an agar-based media that allows for the growth of mycobacteria. We'll also put some onto an LJ slant. This is an egg-based slant. The egg provides lipids for the mycobacteria to grow off of. The green color comes from malachite green, which inhibits gram-positive organisms. One other common media used in the acid-fast bacilli laboratory is the MB bottle, which stands for mycobacteria bottle. This we use for blood specimens, peripheral blood as well as bone marrow. This bottle is loaded onto an instrument and uses a carbon dioxide sensor to check for growth. So if there is mycobacteria present, they'll consume the oxygen and produce CO2, and that's what's flagged. Once the bottle has been flagged, we'll once again stain it, and if we do see acid-fast bacilli, that's when we plant it onto our other 3 media, the blood agar plate, 7H11, and the Lowenstein Jensen.